I am a biology professor here at UNR, and I'll talk about how I got there. Okay, so a little bit about me. I was born in Salt Lake City, Utah, born and raised. Here's a pretty picture of Salt Lake City, Utah. I was a first-generation college student. So nobody in my family had ever gone to college. I now have a couple cousins that have gone. I was the oldest of all of my cousins, and there's a lot. And I'm the oldest, and I'm the first one that ever went to college. My parents, aunts, uncles, grandparents, nobody had gone. The reason that I bring that up is a lot of times there's people that are first-generation college students, and they get, there's a program here on the UNR campus for first-generation college students because it is a little bit more intimidating. You're not sure, how does college work? How do I register for college? I mention this because it kind of helps explain my personality, I think. I'm very outgoing, which I think helps me be a better professor. But my family, very, very hardworking blue-collar family. There wasn't a lot of emphasis placed on, you should go to college. In fact, my parents were confused. They kept saying, what do you want to prove by going to college? Why don't you just get a job? And I was like, it's better to go to college. And they weren't mean, they were very loving. It was just, they didn't really understand. And nobody's scientific. So in my family, we didn't walk around talking science and all of that. So I just kind of set out on my own. Some early things about me when I was a little kid. I get asked that all the time. Did you know you wanted to go into science? Of course I didn't know. I didn't really, I thought sciencey things were cool. But I really was not. I used to tell people I might be a doctor just because it sounded kind of cool, right? I did enjoy learning about math and science. I wasn't a perfect student. So nowadays, everybody's like, you got to be a perfect student. I wasn't a perfect student. I was a pretty good one. So I was involved in a lot of normal activities. Yep, I did cheerleading. Yay! Again, I think that helps me be a better professor because I really can cheer on biology and whatever. It was a lot of fun, ran track, did all those types of normal things. So here's one weird thing. There were a few things I used to do that if I think back now, probably were out of the ordinary and might have given me a clue that I was interested in science more than other things. I had an obsession with carp. I've never done anything with fish, but I had this thing in my head that these carp, my parents used to call them the dirty fish, okay? Because they lived in this dirty canal and ditch, and there were a ton of them. And they would always say that, they live in the dirty water. They're the dirty fish. So I got on this mission, and I did it 2 years in a row, that I was going to save all the carp from the dirty water. So and my mom does have pictures of this, I went and got 5-gallon buckets and lined them up in my backyard, and the ditch ran behind my house, and I filled them all up with fresh water. Fresh, clean hose water, right? Because in my opinion, the fish would be much happier in that than dirty canal water. So the fish would swim down the ditch behind my house. I would straddle the ditch like this, and I would grab them with my bare hands, and I would put them in the freshwater buckets. Then I used to give them random things that I thought they might like to eat, like lettuce. And anyway, it was very weird. And it was one of those things that is well documented in my family. One time I had 57 fish that I was trying to save and convert to fresh water to make their life better. And they all died, of course, because they were in a little 5-gallon bucket anyway. So they all died, and my parents would explain to me they can't live in clean water. I didn't believe it. Anyway, I did some really weird things like that. I used to also capture mice. Yes, as a little kid. Anyway, whatever. But I would get mice and I would put them in shoe boxes, poke holes in the shoebox so they could breathe, and give them cheese. So they were in a box with holes poked in it with cheese in there. And cartoons, they always ate cheese. So I thought, and then of course the mice would die too. So a lot of failed attempts at science as a kid, but I guess some good indications that, none of my siblings did anything like that. So, okay, weird things about me. All right, so then I went to college. So graduated high school, decided to go to college, wanted to go to the university. So up to the University of Utah. At this point, I was pretty sure I might want to be a medical doctor based on the fact that I'd had my appendix out and a wrist surgery. I was really curious, really obsessed with the human body. So I thought, yeah, I think I want to be a medical doctor. So I started off as pre-med, which is not a major, okay? When you go, it's not here at UNR either. You declare, you could major in, history, but you just have to do the medical school prerequisites. So I decided to major in human biology. At University of Utah, their biology department split into different emphases, so you have to pick one. So human biology, which meant I took a lot of human anatomy and physiology type courses, okay? So I had a really strong background in anatomy and physiology during my undergraduate. So one thing that happened while I was an undergraduate, which is why I'm telling you guys a lot of times I meet with students that are unsure what they want to do. Because as you're working on an undergraduate degree, a lot of times you'll take a class that just blows you away. And it's something that you had no idea you would be that interested in, okay? And I was doing human biology. I was dissecting cadavers, all of this, and I loved it. Well, I was required to take an elective course outside of human biology, but it had to be within biology. So I took a mammalogy course that focused on deserts and had a field trip component. And we learned all of these mammals, all this stuff about mammals, desert mammals especially. And then we went on a 3-day field trip to the desert, to the Mojave Desert actually. And we trapped rodents. We went around, took photos of different mammals, and it was amazing. I absolutely loved it. And I remember talking to the professor, and he was like, well, if you're a grad student, then you can do this and this with this class. And at that point in my career, I didn't even know what a master's degree was or a PhD. I knew that they were called doctors because they had a PhD, but I didn't know the process. I knew what you needed to do to go to medical school. So I really got interested in figuring out the difference. And I took the MCAT, which I did really well on. And then I decided, okay, I'm going to apply to graduate school, do a year. If I hate it, I'll apply to med school because your MCATs are good for 3 years. So I sort of was hedging all my bets. Yay. Okay, so then here I am at graduate school. I came here to University of Nevada to work with Dr. Steve Vanderwall, who is a world expert on seed dispersal, okay? So the reason I decided to do that is a lot of the plants and animals that I had learned about at University of Utah are also found around Reno, okay? And it's always a good idea to kind of mix up schools if you can, but I knew the organisms and I really wanted to study these. So I came here and started doing some desert ecology research on seed dispersal, which I'm going to tell you guys about. Imagine something like you have an area somewhere where there's a fire. Okay, we experience fires around here. You guys know that. So there's some area, say in the forest or out in the desert or somewhere where a fire moves through, burns everything, right? So then somehow plants get back into that area and grow. And you don't really think about, okay, how does that happen? But sometimes it happens because certain seeds are blown in by the wind. But around here, there are a lot of plants that are dispersed by animals, okay? So here's a pinyon pine cone. Pinyon pine, if you've ever been up Virginia City, you guys know all the pine trees around there, that's all pinyon pine. It's a really common desert tree in the Great Basin. We're in the Great Basin right here. Okay, so here's the seeds. How many of you guys have eaten pine nuts before? Okay, that's what these are. Okay, the pine nuts that you buy at the store are from Pinyon Pine. Really good. Okay, well, the animals love them too. So there's birds, there's chipmunks, different animals that will take these seeds and they want to eat them. Okay, they only ripen in the fall. So there's this huge cone crop. If you've ever gone out pine nut hunting, you go out in the fall, you can collect a ton of pine nuts. All right, well, that's what the animals do. They eat a bunch, but they can't eat all of them, and they want to save some for later. So to save some for later, obviously, we take food home, put it in our pantry. They go around and dig little holes in the soil and hide the seeds from other animals. This is called caching. So they go around and they make little caches all around. Well, most of the time, they'll go dig them up over winter and eat a few of them, unless they're hibernating, and then they'll go do it in the spring, but they always forget some. And the ones they forget can germinate and grow into new plants. So that's how things like burned areas get reestablished, and that's how trees move around. Like if you walk around the forest in the spring, a lot of time you'll see little seedlings coming up. And that is from what's called a rodent cache. Okay, so this is what all my research was on. So this is looking at some pine nuts. Look at this cute guy right here. This is a kangaroo rat. Okay, kangaroo rats live all around here. They're really cool. They're nocturnal. They only come out at night. They hop on their back 2 feet. They put seeds in their pouches, and then they bury them. They're really cool. Okay, so this animal will find some seeds, bury them in a cache, forget to retrieve the seeds, and then if they are in an okay place where they're not going to die when they come up, you'll see little seedlings emerging, and then some of these might reestablish or grow into a new pine tree. Okay? So there are a lot of plants that do that in the desert. Okay, so now we'll talk about, I knew all this, learned all this stuff about seed dispersal. So I decided to do a project on Pinyon Pine. So after my bachelor's degree, I decided to do a master's degree, remember, because if I didn't like it, I was going to apply to med school. So anyway, I ended up liking it, obviously. So I decided to look at rodents and how they do dispersing pinyon pine. Because before I did this, if you looked in any of the books and literature, they said that only birds do it. So I decided I'm going to see if rodents do it. So I built out in the Pine Nut Range, if you know where that is. So this is this location is just east of Carson City. Okay, so went down there, the mountains back there in the middle of nowhere. I built these big enclosures with help. There's wire mesh right here, so rodents can't get in and out. And then I took seeds. You can see these are some pine nuts. I painted them purple. I painted them whatever color that would signify which trial it was. That's why they're painted. Anyway, these were radioactive. Ooh, that was the biggest, scariest thing for me. My PhD advisor, my master's advisor at the time, put on a radioactive label for them. And this was a special radioactive isotope that wouldn't hurt the animals. Okay, biologically inert, which means they eat it, poop it out, and it doesn't stay in their body. But it stays on the seed. So I had the radioactive labeled seeds, and then I would release different rodents into the enclosure. In this case, this is a white-tailed antelope ground squirrel. So release them, the seeds are in here. I had to use this so the birds wouldn't take the seeds. And then I would come back and they would have cached the seeds. So what I did is I dug up caches, collected data on which rodent species made the most caches, how many seeds there were. So this is a cache that was made by a least chipmunk, this guy right here. This cache was made by a Panamint kangaroo rat right here. And this one was made by a ground squirrel. Okay, so it's pretty amazing. They all do this. Pocket mice, deer mice, pinyon mice, all these animals that, like, I would have never thought this, but they all do it. They find seeds and they go bury them, and then a lot of times they forget about them. So that was pretty cool. So then I wrote a paper and got published saying that rodents, in fact, do disperse pinyon pine, and that they're really good at it. They're actually better at it than birds. So yay, that was a big moment. Then I decided, okay, now I'm going to be the doctor that I always said I was going to be, but I'm going to get a PhD instead, which is pretty awesome. So I sort of went, when you do a master's project can be kind of local and a little smaller scale. If you wanted to get a PhD, you have to really ramp it up, okay? So I decided to look at a whole genus and in North America, and this one was called Ephedra. How many of you guys have heard the word Ephedra before? Like as a drug, like ephedrine. How many of you guys have ever taken Sudafed or something like that? Benadryl. These are all various forms of, you can take ephedrine. It's a drug, it hypes you up, okay? It has a lot of other effects. It's actually banned. That's why it's banned, not just because people make meth out of it, but it's banned because if you take it as an athlete, it raises your blood pressure to dangerous high levels. And I think I have a little slide about that. And a lot of people have died. So it's used as a decongestant. So if you take Sudafed, it's the main ingredient, clears you up, okay? It also increases energy. So if you ever like are feeling sick and you take some decongestant and you feel your heart racing, that's why, okay? But it's banned as a weight loss aid now. Okay, so it's found in this plant that I just showed you guys. Okay, this plant's found worldwide. This is the natural source. Of course, now chemists make it in a lab for drugs, but it was first discovered, like most chemicals are, in some kind of plant. All right, so a little bit about Ephedra. This is really exciting. Any questions so far? I should have brought a Ephedra sample so you guys could see it. Grows all over Reno, but it's so, I mean, if you see it like this, right, you'd walk past that out on some trail by your house or something and not even think anything. It's not like, bam, it's a gymnosperm. It doesn't make flowers. Anyway, I think it's cool. All right, so there's not a lot known about Ephedra except that it has that drug in it, okay, ephedrine and pseudoephedrine. Of course, we do know that Native Americans used to make a tea out of it. So depending on the location, it has different nicknames. One of those names is Mormon tea because in Utah and throughout Nevada, it's also called squaw tea. A different species is called squaw tea because the Native Americans would make a certain type of tea. There is a species in Texas right here. This is called Ephedra antisyphilitica. How many of you guys have heard of the sexually transmitted disease called syphilis? Okay. How many of you guys know that way back in the past, Texas had a bunch of whorehouses? Okay. If you know that, you will know that they used to brew a tea out of this and to try to prevent syphilis. So its actual scientific name is Ephedra antisyphilitica. Okay, so there's a lot of properties in this plant, chemicals in this plant that we've known for a long time, you know, that you can make a tea and that they stop bacteria growth. There's a lot of things that goes on, okay? All right, so 12 species in North America. So I decided I needed to investigate all of them and see how they're dispersed because that's what I had to ramp it up to a big genus. So looking at the different species, these are all the seeds of Ephedra. Look how different they look. Some have these little berry structures, some dry up and you can see the seed, and some have wings. So I did a bunch of studies. I'm not going to go through all the boring details, even though I don't think it's boring. I know to other people it might be. These are all the places that I went to collect seeds. All right. When I went to all these awesome places, a lot of the times I was dragging a husband and 2 kids with me. I won't say dragging my husband because he was really helpful, but we would just all go. It was actually really fun. I flew to Texas several different times and did a bunch of research there. I took my best girlfriend one time, just her and I. It was really fun. So, you know, a lot of adventure going out, but it was all to collect seeds. So you can see it only grows in deserts. And then I did some studies, just like I already told you guys about, like I did for my master's, but this was with Ephedra. Same exact thing. Here's some Ephedra seeds, but I did radioactive seeds and then gave them to rodents. There's my husband who I put to work. Here we were out in the field doing some research, but same thing, rodents made Ephedra caches. And this is from my dissertation presentation, and I put it in here because I love these pictures. So I set up trailmaster cameras with seeds, and then I would get a bunch of cool pictures of the rodents. Here's a right-tailed ground squirrel, kangaroo rat, and another squirrel. You can see this one shoving the seeds in its mouth. So pretty cool. All right, just to prove to you I made my kids work. This is me in Texas collecting seeds. And my husband again, me. And then this is my daughter, and this is my daughter. And one time they had a couple friends come sleep over. And I was like, cheap labor. So I had to plant a bunch of Ephedra seeds to germinate them. So I gave all the kids, these guys are labeling popsicle sticks with which species and how deep they're going to be buried. And these guys, who were like, 2 or 3, here's Dora Explorer shirt, I gave them cups of soil and made them pour it all in. So this is, you know, little scientists in action. They probably won't ever go into science now, but I tried. When it was all said and done, I don't expect you to understand this, but all these ones with the symbols, the reason I'm showing you is just that every species for North America, I was able to ultimately determine the dispersal mechanism. So I published 3 papers out of this, which is a big deal. So I was very pumped, very excited. You know, there's some species in North America that are dispersed by wind, some actually by rodents, which was the main thing I was looking for. And then Antisyphilitica, the one that I showed you guys the picture of in Texas, that has the red berries, is actually dispersed by birds because they eat the berries and then poop out the seeds. So all of that work and 3 publications got me a PhD. And a lot of classes, but you guys know that. Right, so that's what you do. I mean, everybody does something different. That's what I did. So then, after all that, I decided, okay, I want to be a professor. As I was working on my PhD, I was TAing an anatomy lab because I had my human biology degree. Remember how I told you that? So I stepped right in. I did a really good job TAing the lab. So I got done and I applied for a job here. I totally lucked out that they had one because my family was here. My husband had a good job. Everything was set. So I applied for a job as a biology professor here, and I got it. And the main thing they needed me to teach was anatomy and physiology. So that's what I have been doing for 8 years. So I stepped right in, started teaching this class. It's huge, okay? This semester, I teach the second part of the class, 224, and I have 340 students in lecture, and we have about 300 in labs. We have 15 lab sections just for 224. 223 right now has about 330 students. So you can see how big, it's a big class. A lot of students like to take it. Our lab sections are small though. I also lead a dissection team. So I told you guys I got to dissect cadavers when I was in college at University of Utah. So I really wanted to make that opportunity possible for undergraduates here. Most colleges will not allow that anymore, okay? Most universities. There's a lot of regulations. For one, you cannot photograph human cadavers. It's against the rules. It's against the law. It's not just against the rules, right? So in this, period of Snapchat, Instagram, YouTube, whatever, right? Like, you can't go into an anatomy lab and be like, there I am. Send this to my friends, a little selfie, Snapchat. Here I am in the anatomy lab with the cadaver. It's actually illegal, okay? So, huge hoops to jump through. All of the different things that they're worried people might be exposed to. When I was an undergrad, they didn't care about us very much, I guess. I don't know what it was, but it was like, no big deal. Nowadays, everything's regulated. So most universities do not allow cadavers in their undergraduate labs, okay? So you won't ever see them in an anatomy lab. But at UNR, we've really fought to keep this program. So really proud of it. I also teach a desert class, which I'll talk about in a sec. Here's some pictures in our anatomy lab. They actually had some photographers come take pictures, which was funny, but worked out. This is a picture of the lab. You can see we teach a bunch of things. We have a really good program here and a really good acceptance rate to professional schools, so we're proud of that. Our dissection team, as I said, it's very unique to UNR. Okay, he just graduated from dental school, actually. We get cadavers every Friday. We get 4 a year. We dissect them. These are individuals that have donated their body to science to be used for dissection, okay? The UNR Med School has 2,000 people on a donation list. I did not realize it was that many. So it's generally older people that want to donate their whole body. It's not donating organs. So they're aware that when they pass away, their body will go to be dissected for education. It's a huge opportunity, okay? One of the things that we're allowed to do is we're allowed to harvest organs, okay, and keep those organs for education. And that's what you guys will be looking at and touching today. We have a bunch of organs, okay? So what we do is we have our dissection team, which we have 4 of back here. Yay! And they're awesome. And they come in every Friday, and they'll do this for like 5-6 hours. And they dissect and they harvest organs and they work really hard, okay? And they come in and then they teach labs. They help teach labs, and it's really amazing. They do a great job. So when you're back interacting with them, ask them a lot of questions, because this is like what they do and is their passion. And the best thing about my job, hands down 100%, is being able to work with these guys. They are hardworking, they're interested, they're motivated, they volunteer. They're all here on their own time just volunteering because they're awesome and they like to talk to you guys. So yay. All right, here's my other class I teach. Have to show this because, I did this desert thing for PhD and master's degree. So now I teach a small desert class and we go on a field trip. Just like the thing I told you guys that I did that kind of changed my perspective on what I wanted to do, I always talk a few of my pre-med students into taking this class because I think it's great to get exposed to all kinds of things. So that's what college is for. So here's a picture of us on our field trip down in Arizona by some swirl cacti. Yay, very fun. So not a lot of research going on. Right now, I do a little bit of dispersal of Ephedra with a graduate student, and we have undergrads that work with us sometimes. But that's about it. Okay, so here's how this is going to work. You guys can, thank you. So we're going to get out some organs. We obviously can't bring the whole cadavers. They're back in the lab. But we have brain and spinal cord dissections. We have some brains. We have some hearts, livers, etc. If you are not comfortable at all with it, you could stand back, just look at it. I really recommend we brought gloves for all of you guys. So putting on some gloves and just touching things. Touching a brain is amazing. It's not just that it's cool to see what it's in your head, but brain tissue is kind of squishy. You can kind of see why it's so important that you wear that ski helmet or snowboard helmet or bike helmet. You get some kind of big injury on your head and you get a bleed. It displaces your brain tissue. There's a lot of really cool things. Touch a liver. We brought a liver with a gallbladder. Liver tissue is hard, okay? We brought some reproductive organs. We brought a skull. We brought some hearts, some human hearts, one with a pacemaker attached. So one of the things we did is we harvested a cadaver. We get quite a few with pacemakers. Okay, so we've harvested that so you can see how a pacemaker works. These guys will show them to you. You can ask whatever questions. If you're not, you don't have to dive in, but it's a good idea, at least look at them. Even if it's not your passion, you might not ever get another chance to look at a dissected out brain with spinal cord and nerve roots and eyeballs attached. So take the opportunity to at least look at it. It's pretty cool. Okay? All right, so let's head on back there. Like if you were to actually, like say, cut into your arm and see a nerve and you're alive, they're like white and shiny. They're sort of a pinkish white color. Once that something's stored in preservative for a while, it sort of turns a little bit more brown. And a lot of places, your skull really is thick. Like when people say you have a thick skull, yeah. And I mean, and the other thing is we're all a little different. So some people, like when we do these harvests, some skulls really are a lot thicker than other people's, which is crazy. It's always interesting to see how different each human body is once we get in there. Yeah. Every time your heart beats, the electric signal starts right here and then spreads across the heart. And if it goes out, which it does, obviously a lot, because we get a lot of pacemakers. My grandma had one. When it goes out, they put in a fake one. So they put wires, and you can see they go in one of the vessels. So here's the vena cava. They put it in there and they insert it into the muscle wall. And then this, since this won't start on its own anymore, this sends an electric impulse. We order a bunch of these for the actual students in the class to cut open because they have to learn all the chambers, all the structures in the heart and how they all work. And so they have to be able to identify them on a real heart, not just like on a model. The left ventricle is super thick because this is where blood gets pumped to your entire body. So this is your oxygenated blood would be in here. Your left ventricle, see how thick the muscle is? It contracts and forces blood up through your aorta, which is this thing. And then the right ventricle just pumps it to the lungs. So see how like small and it's more thin? Because it just kind of has to go. It has to pump blood from here to here. Yeah. The back side of the eye is actually just like a nerve. So your retina, you've probably heard that word. This light comes in and your retina turns the light into an electrical signal and it sends back to your brain. And then the back part of your brain right here, called your occipital lobe, makes a picture. But yeah, all of this tissue, I mean, it just makes bioelectricity. We have our own electric system in our body. If you have a neck injury, so people that are severely like paraplegic, maybe they have a neck injury, then they lose function of their arms, everything. So, yes. So this is where like these go down through the shoulders and then out through your arms.